Guys, this is the diagrammatic representation of the microspore formation. So I take the anther in dissection. When we can cut and dissect the anther, it's look like this. These are the lobes. These are two lobes, bilobe, and each lobe contains two pollen sacs, so called as thecus. So commonly called bilobed, anther is bilobed and dithecus. That's why they are called as dithecus, because each lobe contains two thecus. So this is the outermost layer called epidermis. This is the inner layer endothecium, middle layer and in middle layer, then after it tapetum. Innermost layer is called tapetum. You know the tapetum plays a very vital role in the process of the microspore, because it provides the nourishment to the developing microspores, and also helping the differentiate of the microspores. So the outermost layer of the pollen grain, that is exine, is also made by the tapetum, that is sporopollenin is released by the tapetum. So these are the some functions of the tapetum layer. So I take the one sporogenous tissues which are in diploid number. When they are differentiate, and then they are starting developing to the microspores. So actually these are in diploid number, means diploid number means 2n number. The chromosomes are the nucleus are in diploid. So this one is the sporogenous tissues. Microspore mother cell also called, or the pollen mother cell, we also called as pollen mother cell. And it undergoes meiosis first. When the meiosis is going on, then the reduction, that means the nucleus reduced to the half number of the chromosomes. So it's two nucleus showing on this structure, and then after it undergoes another meiosis second, then it forms a tetrad structure, means four nuclei are formed, four microspores are formed inside this. Then these are all tetrad are in haploid number of chromosomes. So microspore haploid, we can take only one out of these, and these are all haploid number, means the nucleus are in haploid number of chromosomes are present. So the further what's happened, the vacuole are present which are large in size, and one side is nucleus. So this undergoes a mitosis first. Mitosis, the meiosis and mitosis. Mitosis is not the division of the nucleus, but it's the division of the cytoplasma. So unequal cytokinesis and equal karyokinesis. What's happening to this? In mitosis, there is no reduction of the nucleus, but they are divided equally. But unequal cytokinesis, that means cytokinesis means division of the cytoplasma, kinesis means division, cyto means cytoplasma, unequal means not equally divided. Cytoplasma is not divided equal in parts, and in karyokinesis means nucleus division which are in equally divided into two parts. So in mitosis it's happen. So what's the product we get here? These two nucleus which are in haploid, and these are called as the generative cell, and this one is the vegetative cell. This is the large in size, and these are in the smaller size. So this further going on the mitosis second, what's happen further? Here two generative cells which are called as the sperm nucleus, and this one is the vegetative nucleus. And vegetative nucleus actually, and this are the smaller in size, but the nucleus size is smaller, but the cell size is very large. And the sperm nucleus cell size is smaller, but the nucleus size is larger. So this is the difference, vegetative cell and the generative cell. So what's happened further? It's all the process is carrying on inside the anther. So this microspore is ready to going on the another flower. This process is called the pollen pistil interaction. So when they are attached on the stigma of another flower, this process is going on. The inside of the intine, the inner layer of the microspore, that is intine, it goes larger, and down to the ovary, down to the pistil. This one is the style. this one is the stigma portion, this one is the style, this one is the ovule. This is the female part. The microspores are coming here on the stigma, and when the microspore attached on the stigma, then it start these reactions. The pollen tube is larger in size. The tip of the pollen tube cell contains that calls tube nucleus and two sperm nucleus, and this loops like structure which are showing here, these are called as the callus plug. What is the function of these? These plays important role, not reverse back of the cytoplasma, and these nuclei, the major role plays here. These actually act as a locked function. So this one is the cytoplasma. So further function is going inside the female part. So the fertilization is going inside the ovule, especially embryo sac. Then after double fertilization, triple fusion, and then after endosperm, zygote. So the further process is going on here, but the microspore function here. So this is the overview of the microspore, how it generate and how it reach to the stigma and further function. Thank you so much for watching my video. Please share, subscribe and like.